Welcome to your lecture for Chapter 16, the respiratory system. We've done some of these lectures in lab, or some of these structures in lab, so hopefully some of this is already familiar to you. So just to start with an introduction of the respiratory system, remember its main job is to allow for the exchange of oxygen and carbon dioxide. Oxygen coming from the atmosphere into our bodies and us exhaling carbon dioxide out. Because carbon dioxide is that waste product of cellular respiration. So again, the respiratory system obtaining oxygen from the atmosphere. There are tubes that are there to filter, warm, and moisturize the incoming air, taking it into those gas exchange areas, the alveolar sacs in the lungs. It also helps to remove particles from incoming air, again, regulates the temperature and water content to make sure that it's body temperature and fully humid. It provides vocal sound. My ability to speak now is from the respiratory system. It helps to regulate our blood pH. We'll get more into blood pH. I think it's in chapter 18 when we kind of pair that with the urinary system. But we'll see how it helps to regulate blood pH through the carbon dioxide levels. And in the sense of smell. Smell is very important to us for eating and for just general sensory awareness. So there are different types of respiration. It's not just breathing in and out. There is external respiration, and that is ventilation. That is the actual inhale, exhale. So that process of taking air from the outside in and from inside out, that is your external respiration. Internal respiration, this is at your body cells. So this is when oxygen is being transported into your body tissues through your capillaries, and then carbon dioxide coming back out. And then cellular respiration occurs as an intracellular process. This is when your cells actually take that oxygen that they've been receiving and use that to make energy. We can look at the structural divisions of the respiratory system. There is the upper and lower respiratory tract. The upper respiratory tract being the nose, nasal cavity, sinuses, your pharynx, and your larynx. So your throat all the way down to the laryngopharynx, which is your voice box. The lower respiratory tract is below the larynx. It's the trachea, that tube that you can feel in the front of your throat, the bronchial tree, and the lungs. The bronchial tree being those bronchi, those tubes that are transporting air. Here you can see a nice little depiction of that. Again, hopefully you should be familiar with this already. So let's start with the nose. I'm starting at the upper respiratory tract. We'll work our way down. So the nose is mostly bone and cartilage, just kind of cartilage with some epithelial tissue wrapping it. It has the nostrils that provide opening for the entrance and exit of air, so you can breathe in and out through the nostrils. And they also have these hairs, they're called vibrissae. And this is that first kind of filtering system, so that way as you go to breathe in, stuff can get caught in there, it can get trapped in your mucous membranes, so that way it does not enter any other part of your respiratory tract. Posterior to the nostrils, you have that nasal cavity. It's that hollow space posterior there. It's divided by the nasal septum. It has bone and cartilage, again, as the main tissues that we're seeing. It also has these nasal concha. These are those bones that we talked about that are on the sides of your nasal cavity, on the medial border along that ethmoid bone, that are there to kind of swirl air, so that way as you inhale it gets caught in those swirls and it actually slows it down a little bit, so that way it can be more humidified and cleansed. The conchi are supporting those mucous membranes to increase the surface area as well, so that way because the conchi are round instead of flat, it allows more space for these mucus cells to be able to warm the air and humidify the air. Those mucus cells have ciliated epithelium, and so the goblet cells are there to secrete mucus, but the cilia are there to help trap and move along any particulate matter. A lot of times, if you're not sneezing them out, then if they get swallowed, they get carried down into the stomach, and the gastric juice can destroy any of those microorganisms that were trapped. Again, here you can just see the nasal cavity, those concha, that's these ridges that we're seeing. Here is that epithelium. You have your pseudostratified ciliated epithelium with these goblet cells interspersed that are producing mucus and kind of create this nice layer over top of those cells. All right, so beside the nose, the paranasal, beside the nose, right? Paranasal sinuses. These are in your maxillary bone, frontal, ethmoid, and sphenoid bones. These are hollow cavities, hollow spaces. They're there to help really reduce the weight of the skull and allow this resonance, right? Think about when you're stopped up or you have a head cold and you kind of sound like this. It's because those spaces, these sinuses, are being filled with fluid, and so sound can't reverberate. And really, again, it's this reducing the weight of the skull. If these were fully solid, our heads would be even heavier than they already are. If you go down, we can get into the pharynx. Again, that's the throat. There are different regions, the naso, oro, and laryngopharynx. This is the space behind the oral and nasal cavities, a common passageway for air and food from the nasal and oral cavities. Even though you probably do not consume much food uh, through the nose, it is definitely for air. And then it helps to produce sounds for speech by the passage of air by the reverberation of the vocal cords. And again, those three subdivisions of it. So then you're in your larynx. So the larynx is the voice box. It's that protrusion that's kind of midway down the front of the throat. It's that enlargement of the airway superior to the trachea. It transports air in and out of the trachea, kind of acts as this gatekeeper for it. Helps keep particles from entering the trachea, right? That epiglottis can close over so that way food and things don't fall down into the trachea. It also houses the vocal cords that are actually there for producing sound. It's mostly a framework of muscles and cartilage with some elastic tissue. The three big pieces of cartilage that we should look at are the thyroid cartilage, cricoid cartilage, and epiglottic cartilage. So the thyroid cartilage forms this large shield on the front of it. The little protrusion right here in the front, the laryngeal prominence, that's what they'll also sometimes call the Adam's apple. On males, it tends to be a little bit thicker and more pronounced because of testosterone. It just makes that cartilage tissue a little bit more pronounced. You have the cricoid cartilage, which is helping to anchor the thyroid cartilage to this tracheal structure below it. And then you can see here, posterior view, this epiglottic cartilage, and this is what will cover the glottis, the opening to the trachea when we swallow. So inside of the larynx, we do have two pairs of folds, of vocal folds. In the lecture, we call them vocal folds, the true and the false. I believe in your lab manual, it talks about vestibular folds and vocal folds. So I'll talk about that in a moment. But the two vocal folds, they have muscle and connective tissue covered with a mucous membrane. That's pretty common throughout the respiratory tract, is this mucous membrane is pretty much lining the whole thing. So the muscles are there to help move this connective tissue to adjust the shape and the opening of the connective tissue to adjust sound production. So the upper pair of folds of the vocal folds are the false vocal folds. These are not involved in sound production. These are the vestibular folds. Is how your lab manual records those. The lower pair are the true vocal folds, or true vocal cords that are involved in sound production, which I'm having trouble with today. So the lower pair, those true vocal cords, are the ones that are going to be moving and adjusting to produce sound. The false vocal cords, they kind of just act as this covering. They kind of act as like a shielding layer over the true vocal cords. If you combine the true vocal cords with the opening between them, you get the glottis, the opening that is going to lead into the trachea. This is the glottis. And so this is what's really kind of being adjusted when we're producing sound. While we're breathing, those vocal cords relax and the glottis stays open, so that way air can be moving easily. Air can be forced through the glottis. It causes it to vibrate, and that's what produces sound. Changing tension on those cords controls the pitch of the sound. I can move it up and down depending on how much tension gets put on those. The loudness of the sound depends on how much air you're forcing through. So if you don't force very much air through, the sound can be softer. And if you do force more air through, the sound gets louder. The pharynx, oral cavity, tongue, and lips turn that sound into words. You need all these structures to be working in conjunction with each other to be able to produce sounds that actually have meaning for us. Again, epiglottic cartilage making up the epiglottis. That should be pretty easy connection there. Usually it's going to be standing vertically, allowing air to be moving in and out. So the resting state for all of these structures, you notice, is patent. It is open, so air can be moving freely. You are breathing a lot more than you're eating. Uh, Sorry, I just finished eating lunch, and so I was just kind of thinking, I was like, well, you were eating a lot, but you are breathing more than you're eating, and so that the standard state is for those things to be open. But as we do swallow, those false vocal cords and epiglottis close off and prevent things from entering into the larynx. Here you can see the structure when they're closed, when you have the tension on the cords closing the glottis versus when it is open and patent. And then yes, there you can see an actual picture of that. That's the trachea right there. There are your glottal folds, your vocal folds, and then the vestibular folds, or the false vocal cords above it. All right, once we move through the larynx, we're now in our trachea. This is the windpipe. This is just a cartilaginous tube. It is always open. It has some cartilaginous rings that wrap around the front of it that help to keep it nice and open all the time. Really, again, it's just going to be the structures in the larynx that control movement in and out through the trachea. It does sit anterior to the esophagus. The esophagus is just behind this, and it kind of stays closed. It's one of the reasons why it's not, the cartilaginous rings are C-shaped and not O-shaped. If they were O-shaped, then the esophagus wouldn't be able to open when we swallow. So by having this kind of softer posterior region to them, the incomplete cartilaginous rings allows the esophagus to expand. As you go down the trachea, it starts to split into the primary bronchi. And right at that split is what's called the carina. This is that receptor, that irritant receptor. So if particulate matter gets down into your lungs or gets down into the trachea, this can be activated for us to cough out that material. The inner wall, again, ciliated mucous membrane, a lot of goblet cells, so a lot of mucus, a lot of cilia to help keep material moving back up the respiratory tract. Yeah. So here you can see just kind of this basic picture of the lower respiratory tract, the larynx and the trachea, where it splits into the two bronchi, and then they go into the lungs. So let's look at that a little more. This bronchial tree, so we call it a bronchial tree because if you were to hold it upside down, like with the trachea being the trunk, you would see all these branching bronchi that come off of it. As it is, the trachea, is the superior portion, and then the bronchi spread out from those. Those bronchi keep dividing until they get microscopic and connect to the alveoli. The three first ones, the primary, secondary, and tertiary bronchi, also main, lobar, segmental, whichever one you want to use is fine. I like primary, secondary, tertiary. It's just much more descriptive. Primary, obviously the first branch. Each of those bronchi will lead into a lung. So you have two primary bronchi. And then you have the lobar bronchi. There are 5 of these. These each enter a lobe. So on the right side, you have 3 lobar bronchi, and on the left side, you only have 2 of the lobar bronchi. And then you get into the segments. Each lung has about 16 to 18 segments in it, and so each of these tertiary bronchi are going to be going into one of those segments, feeding it. They continue to branch and divide until you get into things like the bronchioles, small organs off that segmental branch. Then you have terminal bronchioles and respiratory bronchioles. So terminal bronchioles are the smallest that are conducting air without gas exchange. The respiratory bronchioles have alveoli, so they actually can perform gas exchange. So terminal bronchioles are the last ones that are just air passage. Respiratory bronchioles are actually involved in some type of air exchange. From there, then you get into things like the alveolar ducts, the sacs, and then the individual alveoli. And so this is where the actual site of gas exchange is happening between air and blood. These alveoli are surrounded, wrapped around by capillary beds, so that allows that very close contact for carbon dioxide and oxygen exchange. Here you can see this picture, again, should be something pretty familiar for you. As you go all the way down, you can pull it out and you can see the terminal bronchiole, that leads to a respiratory bronchiole, and then you get into your alveolar sacs and individual alveoli. And there they are wrapped in the capillary beds. Again, you can see how nice and close contact they make. So this really, the more space they can reduce, so the closer in contact the capillary and alveoli can be, the better the gas exchange will be. And there's just a micrograph picture of that. All right, let's look at some characteristics of the bronchial tree. So we said the trachea is made up of mostly cartilaginous material, and so that's pretty true as you go through the bronchi. They are mostly cartilage, kind of keeping them nice and open. But as you get smaller and smaller, it becomes more and more absent, and as you lose cartilage, you're actually gaining smooth muscle inside those walls. And so by the time you get to the bronchioles, you have no cartilage. It is almost exclusively smooth muscle that's um, comprising the tissue of these bronchioles. That allows for the changes in diameter of bronchioles. So we do bronchodilation and bronchoconstriction. Bronchodilation is opening up the bronchioles, so they're increasing the diameter. That causes that fight or flight. So sympathetic stimulation causes you to increase your oxygen intake, so you're going to have bronchodilation to take in more air and release more carbon dioxide. Bronchoconstriction, that is when you're decreasing the diameter of the bronchioles. So again, not necessarily parasympathetic stimulation that's going to do that. It's more just the lack of sympathetic stimulation. But it can also happen in response to allergic reactions or asthma. It can cause the bronchoconstriction to close them off because it's the body trying to protect itself, but it's ultimately a self-harm behavior because that can then, of course, reduce air intake. Lining the bronchial tree are these mucous membranes, again, filtering, warming, humidifying incoming air. That should be kind of just when you see mucous membrane, you should think filtering, it's trapping stuff for sure. The alveoli themselves are only simple squamous epithelium. So again, if you remember back to our lecture on the integumentary system or tissues, simple squamous. So these are single layer, very flat cells, optimal for exchange of materials. And so that's why they're able to conduct rapid gas exchange. Here you can see kind of a basic depiction of that. So this sac is obviously the alveoli with the air filled in it. And then these are your vessels coming from the heart, right? This arterial, this deoxygenated arterial. Here's your capillary where they're meeting, and then this oxygenated vein going back to the heart. And you can see oxygen leaving, carbon dioxide coming in at all points. All right, so then let's talk about the lungs. That was kind of following the bronchial tree. The lungs are these organs that are kind of housing and wrapping around all these things. Soft, spongy, cone-shaped organs separated by the mediastinum, enclosed by the diaphragm at the bottom, the thoracic cage around it, and they occupy most of that cavity. You have a primary bronchus and large blood vessels, pulmonary veins, pulmonary arteries entering each lung along the medial surface through that hilum, that entrance point in those lungs. The pleura, so if you remember, the pleura is referring to your lung cavity, and specifically these lung membranes. So you have your visceral pleura that's actually attached to the surface of the lung, and the parietal pleura that's along the wall of the thoracic cavity, up along the bones of the thoracic cavity. In between those two membranes is the pleural cavity filled with serous fluid. So this is, we always say, as a potential space, because really these two membranes are kind of pressed right up against each other. They have this serous fluid in between them, though, that's acting as this lubricant, so they can actually, your lungs can expand and contract, and they don't get stuck to the insides of the lungs or to the inside of the chest wall. It allows this kind of gliding motion to happen. So the pleural cavity, we say, is a potential space because there's really no space in between these two, and if there were space, that could actually cause your lungs to collapse. That's one of the ways that you can get a lung to collapse, is because air gets into this pleural cavity, and that pressure pushes the lungs inward to collapse. Right lung is larger than the left because the left has the heart pointing into it. Right lung with its 3 lobes, and the left only has the 2. Again, as I said, the lobar bronchus supplying each one of those lobes, and then you have air passages for things like alveoli, nerves, blood vessels, lymph vessels, and connective tissues. Here is just a superior view of the lungs, so you can see how they're kind of partitioned and divided, all these blood vessels, respiratory bronchioles and things distributed all throughout them. And then you can see also the pleural membranes. The visceral pleura, that thin green line attached to the lung itself, the parietal pleura lining the chest wall, and then the pleural cavity filled with that serous fluid. This is the heart. These are just a chart with the parts of the respiratory system, their description and function. This is a great thing to study, great thing to learn through. All right, let's look at some mechanics of breathing. So that was kind of most of the structures of the respiratory tract. Now we're getting into how we breathe and that regulation. So breathing, we can call it ventilation. So if someone's on a ventilator, they're being artificially directed to breathe. But ventilation is just the movement of air from outside the body. So this is not internal respiration. This is an external respiration process. Ventilation is both inhalation and exhalation. Inspiration, expiration, whatever you want to call it, it's the same kind of words. Inspiration is moving air from higher to low, rather, all of these are both. This first little bullet is a strange bullet, but it says anyway, air moves from higher to lower pressure. That is true. When you're inspiring, when you're inhaling, air is moving from the higher pressure outside in the atmosphere around you, and then it's moving down into your lungs, which are a lower pressure. Atmospheric pressure is that pressure within the air all around you, exerted on all objects it comes in contact with. So even though you don't really feel air pushing on you right now, if you've ever been in an airplane or you've gone scuba diving or something where you're changing your altitude greatly, then maybe you notice, oh yeah, you can kind of feel that air pushing in and out through your ears. So atmospheric pressure is this pressure, again, that's pushing air into our lungs. Once that air inside the lungs decreases below the atmospheric pressure, if you're not breathing in, your body is maintaining either the same pressure level of pressure. You can breathe out where you're actually your lungs are higher than air pressure, or when you're breathing in, you're expanding the volume of your lungs, and as you make your lungs bigger, the pressure inside the lungs gets smaller. There's an inverse relationship there between pressure and volume. So as I make my lungs bigger, the air pressure inside the lungs starts to drop, and it drops below atmospheric pressure, and that pulls air down in. You can use muscles like your diaphragm and your external intercostals to do that. Same thing with like the scalenes up in the neck. These are the normal ones, though, that are expanding the thoracic cavity for normal inspiration. So when we say the primary muscle for respiration, your diaphragm, it's because it's pulling down on the base of the lungs to make them bigger and make them expand. Your external intercostal muscles also assist in this by making the lung a little larger. This one is one of those you kind of have to remember it backwards. External, right? So you might think X for exhalation. No, external intercostals for inspiration, internal for expiration. So they're just backwards. Those external intercostals in between the ribs are going to move the sternum and ribs upward and outward to expand the rib cage. And that again results in the pressure in the lungs following below atmospheric pressure, and air rushes in. There are a couple of different ways that you can inhale and exhale. When you're just doing it regularly, just doing it normally, it's just a kind of casual process, but you can force these things. So a maximal forced inspiration, so you're taking a deep breath in. You're using more muscles, pectoralis major, sternocleidomastoid, scalenes, all of these things to help pick up and expand the rib cage. Because you have that surface tension between the layers of the pleura, the visceral and parietal pleura, the lungs keep expanding with it to make them bigger. Without the surface tension, though, is not advantageous to the alveoli. It would tend to cause them to collapse. So that increase in pressure would cause sometimes the alveoli to collapse down, especially as you exhale. And so surfactant is something that is produced to keep the alveoli inflated, preventing their collapse. So kind of like the serous fluid, creates this surface tension in between the two lungs membranes to keep them open. This surfactant that the alveoli produce kind of helps them stay stuck open as well. So even as you exhale and increase all that pressure, they still stay open for gas exchange. Here's a depiction of that. So you can see as the diaphragm is pulling down, the lungs are expanding and air starts to draw in. And then as the diaphragm contracts and the rib muscles contract, pressure starts to increase inside the lungs. You're decreasing volume, increasing pressure, and so air moves up and out. And this is just showing normal and maximal inspiration. When you're breathing maximally, your sternum starts to lift. These other muscles start getting activated. All right, so expiration, that was inspiration. Expiration is different in that it tends to be a passive process. When you're just exhaling normally, while it takes muscles to inhale and open the lungs a little bit more, them relaxing is a passive process. It's that elastic recoil of the muscles and lungs going back to their original state. all the muscles start to relax, decreasing the volume of the thoracic cavity. And as that happens, it starts to go back to its normal shape. The pressure inside of the lungs starts to increase, and you exhale out. You can also do the maximal forced expiration. So if a normal exhale is a passive process, it's just a passive process. But if I wanted to, I could continue beyond that passive and really force air out. Expiration of more air than normal, you start to use your internal intercostals, right? Internal expiration, as well as your abdominal wall muscles. You actually start to use your abdominals to contract and put even more pressure from below the lungs up and out. This is again just showing normal versus maximal expiration. The lungs shifting here, the diaphragm contracting back in or relaxing back in to its normal shape, which is rounded, right? Passive is just kind of the diaphragm would be rounded rather than flat. And then for that maximal expiration, you have your abdominal muscles, the intercostal muscles, all working to try and reduce the volume of the thoracic cavity. So thinking about these specific volumes now. So we talked about different types of inhale and exhale, right? Inspiration versus maximal inspiration. Same thing about the expiration. So we can actually study the different volume changes within the lungs, and that's a way to determine health in someone's lung capacity. So this is called spirometry. Spirometry is the study of various air volumes moving into and out of the lungs due to different degrees of effort. So remember, with those maximal inspiration, expiration, you were adding an extra effort to get more air movement. A basic respiratory cycle is one inspiration and an expiration. So in and out. There are 4 distinct respiratory volumes that we need to look at: tidal volume, inspiratory reserve, expiratory reserve, and residual volume. Your tidal volume is just the volume of air in a normal average resting cycle. So again, it's when you're not really thinking about it, you're paying attention to other things, your body's just breathing on its own. That little bit of air, it's about 500 mils that you take in and out. That's just, the tidal volume. So this is 500 mils. Your total lung volume is about 6,800 mils. So you can think about this as half a liter versus 6.8 liters is your total volume. The inspiratory reserve, this is the volume of air that you can breathe in above your tidal volume. So tidal volume being about half a liter that you can just breathe in and out on normal. But if you wanted to really take in a full breath of air, you can usually breathe in about 3 liters of air. So about 2.5 liters more than just your regular normal resting breath. But again, it takes a lot more effort. You have to use a lot more respiratory muscles for that. Same thing with expiratory reserve. This is how much air you can push out after you've just done your normal breath. So if you just do your normal breath in and out, and then you just keep pushing air out, right? The maximal forced expiration, you end up with about 1.2 liters left. I'm sorry, that the average is about 1.2 liters of how much you can actually push out. Residual volume, this is the air that remains in the lungs after maximal expiration, so 1.2 liters. You cannot measure this with a spirometer because it just sits in the lungs. It sits in the alveoli. It is not accessible. So your lungs are always going to remain inflated as long as they're healthy and normal because they have this residual volume. There's only so much that you can actually push out because this residual volume is just going to stay in there to keep the lungs from deflating. So those were the volumes, the individual movements of air. We can talk about capacities now. So this is combining 2 or more of those volumes. Your inspiratory capacity, this is how much total air you can breathe in completely. So that's your inspiratory reserve volume, that maximal effort, and then your tidal volume, your normal in and out, 3,500 milliliters, 3.5 liters. So we're just kind of basically going through and we're combining these 4 volumes in different ways to talk about different capacities. So if you ever go to a pulmonologist, they would have you do all these different breathing exercises, and they would measure the volume of air that's moving in and out, and they can use that to determine your lung function. Functional residual capacity. This is volume of air that remains in the lungs after a resting expiration. So just a resting expiration means a passive one that you're not forcing, so not the maximal effort. Vital capacity, maximum volume of air that can be exhaled after a maximal inspiration. So you breathe in as much as you possibly can, and then you breathe out as much as you can. Total lung capacity is all of that. It varies with age, gender, body size. It's the total amount of volume the lungs can hold. I think I said 6.8 before, 5.8 is what I meant to say. Sorry about that. So total lung capacity is all of the air that you can hold, so your vital capacity plus your reserve volume. There's also this volume called anatomic dead space. And this is a volume of air that's in the bronchial tree, not involved in gas exchange. Because when we're talking about breathing in and out, as you inhale, air comes down through the trachea, it goes into the bronchial trees, but some of that air is going to stay in those bronchial trees. It'll push older air into the alveoli for their gas exchange to occur, and then as they exhale, they might kind of slide past each other. But it's not like every inhale exhale is completely filling your lungs and completely emptying your lungs. As you can see with these capacities and volumes, there's always some amount of air that's being left over. And a lot of times there's this air as well in the bronchial trees that is not really being involved in gas exchange at that point. It's just kind of sitting there in those passageways because there's only so many places for air to go into your lungs. This is a lovely, not confusing at all chart that shows the different respiratory volumes and capacities. So this one, it does look very confusing, especially if you're still not quite familiar with the different volumes and capacities. So volumes over on the left-hand side, capacities over here on the right-hand side. And just remember, capacities are different combinations of your volumes. That's why you got these multicolored arrows. And then a nice little chart if you like to see it written out instead. Okay, there are also other ways you can move air, these non-respiratory movements, so not really used for breathing, more for clearing air passages or sometimes expressing emotion. So coughing and sneezing are the reflexes that are there for clearing the air passages. Coughing is the lower respiratory reflex from the lungs initiated by the carina. Sneezing is one that's clearing your upper respiratory passages. Things like laughing and crying to help express emotions. It's this quick inhale, exhale. Hiccups, that's when your diaphragm starts to spasm, so it's contracting, relaxing really quickly. No real known function of why we hiccup. It is just kind of a glitch in our diaphragm system, basically. And then yawning. Yawning is one of those kind of classic reflexes that they really don't know. There's a lot of theories, but if anybody's telling you, "Oh, I know why we yawn," they don't. There are a lot of different theories. Some of them are wild. And there's really no known cause for why we yawn, but it can help to increase your oxygen intake. So that might be, something if you're needing to do a deeper breath. If your pH is starting to get off or something, you can take a deeper breath through a yawn, and it will increase some more oxygen for you. All right, so thinking about that, look at our control of breathing. So normal breathing, rhythmic, involuntary event. You don't really think about it, even though you do have that ability to be able to take control of your respiratory rate. For the most part, you're not really thinking, it's just doing it on its own. There are some couple different areas in the brainstem that are controlling breathing. You have neurons in the medulla and the pons that can affect the rate and depth of breathing. There is the medullary rhythmicity center. It has a ventral and dorsal respiratory group. So within your medulla oblongata, you have the ventral and dorsal respiratory group. The ventral one stimulating respiratory muscles, basic rhythm of breathing. Dorsal is really controlling the rate and the depth. The pontine, so within the pons of the brainstem, the pontine respiratory group can contribute to rate and depth and limits duration of inspiration. So it's going to limit how much you actually take in at a moment. Here you can see the brainstem with the pontine and then the ventral and dorsal respiratory centers and their connections down to those respiratory muscles. Here's a little flowchart of that. The pontine respiratory group can influence the medullary respiratory centers to change the rhythm and depth of breathing. Right, so what are some of those factors that are affecting breathing? Because we said it can be voluntary or involuntary. Things like chemicals, lung tissue, stretching, your emotional state, and your physical activity all affect your breathing, especially physical activity. As your temperature increases, pH changes, CO2 changes, it can really affect your breath. You have a couple of different chemoreceptors. So chemoreceptors, these are measuring different chemical concentrations in the body. There are central chemoreceptors associated with centers in the medulla oblongata. They're really sensitive to changes in CO2 and hydrogen. We'll see that CO2 and hydrogen play a much larger role in breathing regulation than oxygen does. And these central chemoreceptors are specifically looking at the cerebrospinal fluid. And that's because CSF is representative of blood concentrations, so it's a pretty accurate measure still. So if either of those starts to increase, so you have more CO2, more hydrogen, that means your pH is decreasing. Your blood is getting more acidic. And so those central chemoreceptors are going to signal an increase in rate and depth of breathing to help bring the pH back into balance. It does that because the more you exhale, most CO2, 70% of carbon dioxide, well, all your carbon dioxide is transported through your blood. And so as you exhale, you're getting rid of that excess CO2. Blood and CSF levels start to fall, pH comes back to normal, and breathing can return to normal. Uh, Breathing rate starts to decrease if these concentrations get too low. So let's say you're breathing too fast, you're getting rid of too much carbon dioxide, your body will start to send signals to lower your CO2, lower your breathing rate to help bring CO2 back into that normal range. Blood oxygen, as I said, has little effect on it because there is such larger shifts, much bigger shifts in blood oxygen concentration than there is in CO2 concentration. It'd have to be very low, right? Though, like if it does start to really drop 50% below normal level, then those chemoreceptors will start to get stimulated. The other exception to this is if you have a chronic lung condition, something like emphysema, where your CO2 levels are going to be chronically altered, then your chemoreceptors might start monitoring your blood oxygen levels instead because they're going to be more consistent. Those peripheral chemoreceptors, these are in your carotid bodies and aortic bodies. So carotid artery in your neck, aortic body in your aortic arch. They are monitoring oxygen levels, send those signals to the respiratory center to change breathing, but they only have minor control. Really, the central chemoreceptors monitoring CO2 and hydrogen, that's what's really regulating the involuntary breathing process. Here's a nice little flow chart for that. If you decrease your ventilation, decrease your breathing rate, you're going to, oh, sorry, let's start. Oh, no, that's correct. That's correct. Decreasing ventilation, as you decrease your breathing rate, you're not exhaling as much. So you start to have an increase in PCO2. So this is the partial pressure of carbon dioxide. You start to get more CO2 in your blood. That's going to decrease your blood pH, make it more acidic. That's going to trigger those chemoreceptors, and they're going to send signals to your medulla oblongata. That then sends signals to the spinal cord motor neurons. Your respiratory muscles are activated to increase your ventilation, breathe faster. And as you breathe faster, you're exhaling out that carbon dioxide. And what do you know? This is providing negative feedback. Because as PCO2 starts to drop, you start inhibiting these signals, and your ventilation can return to normal. There's just a little depiction of how those things might talk to each other. All right, so other factors for affecting breathing besides just your chemoreceptors. There is the inflation reflex. So this makes sure that you don't overinflate the lungs. It's that, if you ever take a really deep breath, you kind of breathe in, and maybe sometimes you notice you're like, oh, I didn't quite breathe as full as I could. Sometimes I breathe fuller. Then you take a full breath, and it kind of hits a point where you're like, oh, okay, I have to exhale now. That's an inflation reflex. It's baroreceptors, stretch receptors in the visceral pleura, the bronchioles, the alveoli, making sure that you're not overinflating during forceful inhalation. Emotions, fear and pain, those can definitely alter your breathing patterns. If you've ever been scared or in an accident or something, your heart rate starts to increase, your breathing rate starts to increase. It can really shift your state of breathing. Voluntary control over breathing muscles, so you can stop breathing for a short period of time. You can hold your breath. But as CO2 starts to build up, chemoreceptors get stimulated and breathing will resume. So even if it is one of the things you hold your breath till you pass out, you're going to start breathing again as soon as you pass out, as soon as your body is able to take back over. The opposite being hyperventilation. So overventilating yourself, breathing really quickly and deeply, it starts to really lower the carbon dioxide levels in the blood. All right, so let's look now at alveolar gas exchange. So the actual process of how oxygen and carbon dioxide can move across this membrane. So remembering the alveoli are microscopic air sacs at the distal ends of the alveolar ducts at the end of your bronchial tree, only sites of gas exchange between air and the blood. So you have your respiratory bronchial, I'm sorry, that's the interlobular. You have your respiratory bronchial into the ducts, right, and then into these alveoli wrapped in these capillary beds. So gas exchange is happening across the respiratory membrane. The respiratory membrane is consisted of three structures. You have the alveolar structure, the capillary structure, and then what's called a basement membrane that's holding them together. So along the alveolus, it's simple squamous epithelium, right? Single layer of flat cells. Same thing with the capillary, simple squamous. These are the endothelial cells that are making up the wall of the capillary bed. So both of them are very, very small, very, very thin. And like I said, you've got this fused basement membrane of the two layers. So here inside of your alveolus, right, your alveolar duct would be, it's not really being depicted right here, but you can just imagine kind of like a little tube or passage connecting this individual alveolus to the rest of the alveolar sac. You have the respiratory membrane, these three structures. You have the epithelium, the wall of the capillary bed, this basement membrane in between them, and then the wall of the alveolus. So this is when it starts to get a little bit into some serious physiology of this. We're talking about partial pressures and movement of gases across the membrane based on pressure differences. So in a mixture of gas, so in the atmosphere, you have a mixture of oxygen, carbon dioxide, hydrogen, nitrogen. Those are just the big ones too. So you have this mixture of gases, and each gas is going to make up a different concentration. Oxygen is about 21% of total atmospheric air. And so we say its partial pressure is 21%. If you were to go and put, take just normal oxygen, normal atmosphere at sea level, and you were going to take that and put it into a box, and then you put that box inside of a room where the oxygen percentage, let's say inside of the room, was only 10%. Because you have a box containing oxygen at 21%, that's higher than a room filled with 10%. The oxygen in the box is naturally going to leak out into the room because the pressure of oxygen inside of this one container is higher than it is inside of the room. So oxygen is naturally going to want to move to that lower area. So partial pressures are really what drive this process of diffusion. So partial pressure of oxygen is always higher in alveolar air than in capillary blood. So oxygen diffuses into the blood. Right, there's some nice fun little numbers here looking at different pressures. So it's talking about atmospheric pressure, 760 millimeters of mercury at sea level. Air is 21% oxygen. So the percent of oxygen, the partial pressure of oxygen, that's what this P stands for, is partial pressure of oxygen, is 160 millimeters of mercury. If we look at the PCO2 of air, it's of CO2, it's 0.3. So much bigger differences in terms of concentration, for sure. The PCO2 is greater in blood than it is in alveolar air, though. So normal air PCO2 is 0.3. In your blood, it's going to be higher than that because your body is actively producing CO2. And so naturally CO2 is going to want to flow from its higher concentration in the blood to its lower concentration in the alveoli. So moving opposite of the carbon of oxygen. So partial pressures, even though you have, just a mixture of air based on the gas that you're observing, they can move in opposite directions. Because it's not like, as you're seeing in air, there is CO2. It does have a partial pressure, but it's so much less than the PCO2 in blood that CO2 is only going to diffuse into the air, into the alveolus. So in a gas mixture, each gas diffuses from higher pressure to lower partial pressure, dependent on that gas. Factors that can increase gas diffusion: more surface area, so having all those alveoli, those millions of alveoli, have a lot more opportunity for this gas exchange. a shorter distance, so capillary beds being pressed right up against the alveolar sac. Greater solubility of gases, so we'll look at that. Oxygen is very soluble, and so it dissolves pretty easily. And a steeper partial pressure gradient. So the more pure oxygen you're breathing in, the more will enter into your lungs. Here's a nice little depiction of that. So coming from the right ventricle, so from the heart, this pulmonary artery, you can see you have your PCO2 is 45, your PO2 is 40. So fairly low oxygen, pretty I mean, we'll see CO2 only changes about 5 bars here, but it is still higher than in the outside. In the alveolus, which represents the outside air, your PCO2 is 40, so 45 is higher than 40, so carbon dioxide will move out. Your PO2 in normal air is 104 compared to 40 in the capillary, so PO2, your oxygen will move into your capillary, so that by the time you get to the this other end, going back to the heart, your CO2 is back down to 40, and your PO2 is at 104. So then transporting that gas. So once you get this exchange of gas happening, you have oxygen-rich blood again. The respiratory gases have to be transported somehow or dissolved into the plasma. So first, let's look at oxygen transport. Oxygen is not really easily dissolved into our blood, but it is transported on hemoglobin. So 98% of your oxygen is bound to hemoglobin. It's called oxyhemoglobin, and that is how, again, the 98% of your oxygen gets transported to cells. The other 2% just gets dissolved into the plasma. Think about like a soda, right? Sodas, the reason they're fizzy and bubbly is because carbon dioxide gets dissolved into them. They take this gas and they force it into the liquid to make it dissolve. That's kind of what happens with oxygen. The pressure within the blood vessels is so high that it actually forces oxygen to dissolve into your bloodstream. When you breathe normal air, alveolar PO2 is always higher than that of alveolar capillaries, so it's constantly diffusing across that respiratory membrane. Each hemoglobin can carry up to 4 oxygen molecules. They bind to an iron molecule in the center of it, and so they can hold up to 4 of those. Oxyhemoglobin is just hemoglobin with oxygen attached to it. When you're in the lungs, in the capillaries of the lungs, hemoglobin is going to be 100% saturated. It's not really true once you get into the actual heart. It tends to be about 97 to 98% saturation of oxygen is normal. And again, that means that 97 to 98% of your hemoglobin molecules have oxygen bound to them. At the lungs, because the PO2 is so high, then it tends to be about at 100%. Those bonds are pretty unstable when oxygen starts to get low. So there's some other factors that can influence oxygen offloading, but it really kind of makes the hemoglobin change its shape because our bodies need to be able to get rid of that oxygen. If hemoglobin works so that it stayed bound really tightly, that would not be very efficient in energy production. So as oxygen starts to kind of unbind and unload into the tissues, hemoglobin actually makes it easier. It changes its shape, so it makes it easier for it to get rid of that oxygen. Uh, but even in venous blood, hemoglobin is still 75% saturated with oxygen. So that way you have this O2 reserve in case you need it for emergencies, for running or fighting or whatever it may be. So when we talk about deoxygenated blood in your veins, it's still 75% saturated with oxygen. That's again, our bodies would freak out if that's all if that was level that it stayed at, because typically they like to have 97, 98% saturation of oxygen. Things that can increase O2 release from oxyhemoglobin, blood concentration of CO2, that also will inherently increase the acidity of blood, increase in blood temperature. Notice all of these things can happen from increased activity. If you're exercising, if you're running, if you're stressed, all of these things will occur and your body will naturally want to get rid of more, offload more oxygen to the cells for energy production. A deficiency of oxygen reaching tissues is called hypoxia. Lots of different causes for that, but it essentially inhibits the body from getting the oxygen that it needs. Here you can see a nice little circular graph of from the alveoli through circulatory system into the tissue cells. I recommend going through this, looking at all the different partial pressures, the movement of different molecules, both within the tissue cells, within the alveoli to the blood, your external respiration and your internal respiration here, and then seeing how things are transported. So carbon dioxide transport. CO2 transport, most of it tends to be transported as what we call bicarbonate ions. Only about 7% dissolves in blood plasma. 23% binds to hemoglobin. And then again, the vast majority is transported in what we call bicarbonate ions. CO2 acts as an acidic agent, and so it causes your pH to decrease. So once our body is transporting CO2, rather than just transporting it straight as CO2, because CO2 will combine with water to form this carbonic acid, and this is what's going to make your blood pH start to decrease, this carbonic acid. But in the cells, in your red blood cells, carbonic acid will actually split to form this hydrogen ion and this bicarbonate ion. And bicarbonate acts as a buffer. It kind of travels through the blood plasma, so that as this hydrogen ion might be starting to produce more and more, this bicarbonate is there to act as a buffer to keep pH nice and neutral temporarily, because we'll see that this gets exhaled pretty quickly. So CO2 gets transported that way, either bound to hemoglobin, dissolved in the plasma as bicarbonate, right? Just kind of all these different mixtures. Your body finds different ways to transport this waste product, so that way it doesn't really disrupt normal body function. Then once you get back to the lungs, everything gets reversed. Carbon dioxide gets reformed, water gets reformed, and so that's why when you exhale, you're exhaling carbon dioxide and water vapor, because there's a little bit of moisture in your air as you're exhaling from this reaction. Again, CO2 exhaled from the lungs. It's released easily because PCO2 is higher in the alveoli than in the atmosphere. Your body is going to be naturally producing that CO2, increasing those concentrations. And so since CO2 in the air is typically much lower than that in the body, it easily exhales out. Here you can see just a depiction of that. CO2 being produced in the tissue cells, diffusing into the plasma, bound into hemoglobin, and then going through this reaction to produce carbonic acid and eventually bicarbonate, which travels in the plasma to keep pH normal. And then at the lungs, it all reverses. The bicarbonate in the plasma diffuses back into the cell. It gets turned back into CO2, which is going to diffuse into the alveoli. There's this nice little table to help you understand what gases are being transported how, and on what substance. All right. Thank you so much.